We do give a welcome now to Professor Keith Fox, who's from Southampton University, where he's the Professor of Biological Sciences, and he's also the Chairman of Christians in Science. He studied here in Cambridge and then moved to Southampton and lecturer in 1987. Keith is also the Senior Executive Editor of Nucleic Acids Research, which might not mean a lot to you, but I can know that means a lot of work. Okay, editing a major scientific journal really is a huge amount of work, and Keith is particularly involved in DNA research and so forth. But this afternoon he's going to tell us about something a little bit different, and that is about the ethical issues aroused by science at the moment. So over to Keith. Thank you very much.
Thank you, Dennis, and thank you everybody for sticking it through to the end of the afternoon. I'm very conscious that it's only me that stands between you and dinner, and I'll do my best for you. And it's at the end of the day, which is why I've given you a handout, so that you can doze off and still engage me with polite conversation afterwards, as though you did listen to everything I said. I'm also, unlike our previous speakers who've given you answers, I'm going to spend most of this time giving you questions and leave you to work out the answers for yourselves. One other disclaimer is that I am a biochemist, unashamedly a biochemist, and those are people who, as we used to say in my student days, taught chemistry to the biologists, biology to the chemists, and about beer and football amongst themselves. So I'm not a card-carrying biologist as such. I'm certainly not a medic, and I definitely have no training formally in ethics, although this is a subject I've read a considerable amount about. So I thought if we're looking at bioethical issues, I'll pick up a few websites and see what are the bioethical issues today. So here is the Bioethics Education Project, the website for that. It is on the handout, and there you see a very long list of biological ethical issues. And I thought we might touch on just a few of those this afternoon. So I went to another website and found another long list of very different biological issues. This one from the Nuffield Council for Ethics. And again, we might touch on some of those. This isn't a small area. This is something that touches on a lot of contemporary biological sciences. The root of a lot of the ethical questions comes back to this marvellous molecule that I've had the privilege to spend nearly 30 years doing research on, this molecule DNA. And I put its structure up there for you simply to emphasise that in all the ethical discussions we have, we're dealing with a chemical. It's a chemical with some remarkable properties, but nonetheless it is actually a very boring polymer of sugars and nucleotides and phosphates put together. Some fascinating properties, but it is just a chemical. Put it in a test tube and it looks like a piece of tissue paper that's gone through the washing machine by mistake. So in all our conversations, beware of the temptation to deify DNA. It's a molecule, it has fascinating properties. But we're able to do lots of things with DNA. This structure elucidated in 1953 by Watson and Crick. The first cloning experiments, I'll show you in a minute, were done in 1983. See, that is only 26 years ago. We're able to do lots and lots of things with that. And with those things come issues, some of which were only thought of really after the process of manipulating it started. At the top of people's list of issues, nearly always come the one of risk. Not the actual ethics itself, is it right to do it or is it wrong to do it, but what are the consequences of doing it? What are the risks? But along with that comes some actually fairly deep ethical and philosophical questions as we start to manipulate humans, human DNA, animal DNA, plant DNA, bacterial DNA. Some of these are along the lines of what does it mean to be human? When does life begin? What actually is life? We touched on that earlier today. Who owns biological information? Do you own it? Do the companies own it? What actually is natural, as we talk about changing the natural world, and what rights do you and I have, or do animals or bacteria or plants have? So as we raise these issues, it's worth thinking for a few months about some of the ways that we deal with ethical questions generally, and apply these, see if we can apply them to biology. most people, I think, in terms of the way they make ethical decisions, simply muddle through. It's a gut reaction. You make it up on the spur of the moment. I like it, I don't like it. And maybe think their thoughts through afterwards. We're a little bit more principled than that. Many people will make an ethical decision based on a religious principle. If that's hard and fast, there's then no little scope for debate. Somebody who says, the religious book says this, and I stand by that, you may disagree with the religious book, but you can't then argue as to whether it's an ethical principle or not. The tough thing is that although that says right or wrong, that most of the religious books really make no direct reference to anything that's related to molecular biology or genetics or cloning. You might try and read some things back into them. They say lots about what it means to be human. They say lots about our responsibilities in the world, no matter what religion you follow. But they don't say anything about molecular genetics. Lots of people will take what we call a consequentialist view of ethics. What's the outcome? Is it of benefit? Not should I do it or shouldn't I do it, but what happens at the end of the day? We might talk about rights. I have a right to this, and you have a right to something as well. But what happens when our rights clash? And what happens when our rights actually are unfulfilled? We have a right to good health, but there are plenty of diseases that we can't cure, and ultimately all of us will die. We might talk about natural law. Whoops, I've gone past that. Natural law. What is natural function? What is natural? And we can talk about this if you like. The world we see around us is actually not natural. These nicely laid lawns in the college here are beautiful to look at, but they're not natural at all. They're human creations within this environment. Most of agriculture is not natural. Most of the countryside in this country is not natural. It's man influence. And ultimately, some of us make our ethical decisions on what is called the yuck factor. It just seems repugnant to me. I mean, John Bryant has pointed out I'm of the same sort of generation. I can remember the first heart transplant, where people said, you can't possibly do that. How could you walk around with somebody else's heart inside you? Repugnant. And more recently, we've seen examples of facial transplants. And again, it comes back to the yuck factor. How can you possibly take somebody else's image, their visage, and walk around like that? And as technology changes, so our views sometimes on these things change as well. I wonder what you make of this quote from Steve Jones. So he says, when it comes to medical research and medical technology that works very quickly is accepted by the public, and this goes for, I think, the whole of bioethics. Ethicists may not like it, scientists may not like it, but the public, if they believe it works, they will accept it, and legislation will always follow. Ethics has always followed science, it's never led it, and I don't see any reason why genetics is going to be any different. Ethicists would love to tell geneticists what to do, but I'm afraid the geneticists are not going to listen. I don't know what you make of that. It says that we as scientists, or ethicists, we as ethicists are going to follow on from what science has already discovered and not lead that. I present to you also what somebody else has also pointed out, some very basic facts of human life that we maybe try to ignore. Very simple ones is that firstly you had no control over whether you became alive or not. You were not autonomous in that decision. Somebody else made it for you, or it was a complete accident. Once you're alive, you try to stay alive. That's natural with all life forms, really. Once alive, most species have a strong instinct to reproduce. And that all species eventually die, or individuals eventually die. And it's worth keeping those in mind as we look through some of the technologies that can extend life or change life or modify life in all sorts of ways, is that we didn't choose to be here in the first place, and we won't choose when to go, and all of us will die. So some of the general ethical principles we could use are some of the ones that the medics use. We should do no harm. In fact, we should positively strive to do good. Now, those sometimes are not, they're opposed to each other. in order to do good, you have to do harm. In order to do any surgery, there has to be pain. We should have a process of autonomy, where people can choose for themselves. They're in control, as far as possible, of their own destiny. And we have a system of justice where we're not overriding people's natural abilities and natural likes and dislikes. Before we get to some of the more detailed things, we also need to deal with a few of the phrases that come up from time to time. Are we, as we change genetics, as we deal with genetic modifications, are we playing at God? The phrase you'll frequently hear, it suggests two things. It firstly suggests there are boundaries that we shouldn't cross, even though we may not know what those boundaries are. There's a fence around nature that says don't go any further. And also it suggests that we're playing. Right? This is something that's just done for fun. It may not have any useful consequences. It's just, it's a game. And a lot of what I'm going to show you clearly is not a game. This is being serious. There's also the question that's raised about playing with God because we're made in the image of God. And I want to turn it around the other way. If God is a creator and we in any way are made in the image of God, then part of our activity also is to be creative and to discovering things and changing things and ruling the world in a positive sense. Also we find as we look at the whole topic that the phrases that come up in the media are actually not very helpful. So I'll call this media wars. Think of some of these phrases you see. You'll see that the words frankenfoods, genetically modified food. Call it genetically modified food, yes, but frankenfood is putting a spin on that says this is dangerous, it's where we shouldn't have gone. Even the word genetics to people who aren't familiar with the scientific discipline brings with it all sorts of connotations of eugenics, of something that's unpleasant, it's sterile and clinical. And even the word more so, genetic engineering, brings with that the idea of an engineering principle, quality control, predictable outcomes, quantifiable standards of measurement, efficiency, utility, not the sort of things that we put usually with a biological living human realm. So beware, I think, in any discussion of bioethics, use any of those terms very carefully, because all of those terms, I think, are loaded. So as we look at genetic modification, first of all, we're able to do that because this wonderful molecule DNA is the genetic material for all living organisms. Because it's common to all and the coding is the same for all organisms, it's possible to move it around from one organism to another one, from bacteria to plants to animals to humans. And that a gene expressed in a bacterium could be expressed in a human, and vice versa, human genes expressed in bacteria, because the wonder of DNA is it's universal in its code. So I'll look a little bit at some of those issues as we go through, and I'll look also at some things to do with stem cells and cloning, and ask one or two questions about those, because those are often the issues that come to the fore, and sometimes the more difficult ones to deal with. Firstly, a little history of cloning. It was in 1973, really, that the first genetic modification of a bacterium, E. coli, was done. And it wasn't long after that actually the scientific community woke up to the possibility that this could bring with it substantial risks. I emphasise risks. That's not saying asking a question of right or wrong, but actually addressing whether there could be some things that will be harmful. And it was in 1975 that a conference held in California of senior scientists in the discipline essentially decided to put a halt on genetic modification until they thought through some of the issues. So it was what's been called a landmark of social responsibility and self-governance by scientists. After that pause, things then started to move forward very rapidly. In 1976, we saw the first genetic modification of animals, and actually plant modification came later. That wasn't until 1983. Now, the whole of this area of genetic modification and the rightness and wrongness of it really brings with it a whole list of questions. The ones above the line that I've put there are ones to do, I think, with risk. What are the long-term effects on the environment? What are the risks associated to health? And all the other ones are actually to do with more ethical principles. What about species? Does it blur boundaries between things? Are there controls that should be placed on it? What about the pain and suffering that's involved? What about if we make human traits into animals? Are we using animals as commodities? All those sorts of questions that emerge subsequently, they're nothing to do with risk, but to do with the moral rights or wrongs. So at the start, some people will say that genetic modification itself is ethically wrong because we're crossing species. We're taking something out of one species and putting it into another one. And that is something that is God-given. Species are established and not to be crossed in that sort of way. Genes are part of, if you like, the web of life, and they cannot be looked at in any form of reduction, or rather, they shouldn't be looked at in a reductionist sort of way without disturbing what we call the balance of nature. They're an essential part of the organism's being, and to move it around is an offence to the particular organism. So that nature is constructed according to various kinds, and that we shouldn't blur the boundaries between those kinds. I think from all that we've seen over the past day in the lectures, we'll say that actually the boundaries between species are far from fixed anyway. It's not obvious to a biologist. They'll spend ages debating sometimes what is one species and what is another one. Those things are very, very fluid. But let's look then at some of the applications. Genetic modification and its uses, not just for playing for fun. Pharmaceuticals. Fantastic use of genetic modification in producing pharmaceutical agents. Right way through from insulin, produced in bacteria now, not a human product. Growth hormone, Herceptin, that's the agent used for treating breast cancer, which there's been a considerable debate on the use of that, you may have seen in the media. Interferon, used as an antiviral agent and sometimes as an anti-cancer drug. Those things are ones that are human genes expressed in non-human species now. So fantastic advances and uses for pharmaceuticals. Of course, the uses bring with them the possibility of misuses. So growth hormone can be used in enhancement in various sports. So think through, every time we do something as a positive benefit, of course there's a possibility of a negative spin-off from that, as we, by our very human nature, do things that we shouldn't do with nice things. Uses in agriculture. GM crops, a major debate over the risks of genetic modification of plants and crops. Look at the positive side for a moment, things like golden rice. That's a form of rice that's been introduced that makes extra vitamin A as a dietary supplement. So it's a more nutritious form of rice. You can have rice that's drought resistant, or other species that are resistant to pesticides or herbicides. These things change the way in which agriculture works. The various risks that go with that, though, containment. Is it possible that some of those genes that give resistance to herbicides or pesticides could escape, and we get then the generation of superweeds? All the biological data suggests that doesn't happen, is that these things are kept under tight control, and that when they're put out of their selective environment of being in the agricultural field and get out in the wild, there's no selective pressure and the genes are lost. There are questions about biodiversity, of what are we doing to the diversity of plant life. And I think that's a very real question, except somebody has also suggested that what are garden centres doing to biodiversity? They're probably a greater danger to biodiversity than any genetically modified crop, as we bring all sorts of plants across the globe and plant them in our gardens because they happen to look nice. And there is the question of toxicity or food safety. But like any food, it's got to be properly tested before it's allowed out onto the market. And plenty of genetically modified foods have been used in the US for a long time with no evidence of any toxicity. And nonetheless, if we allow people to be autonomous, what do people think? What are their perceptions? Well, in this country, it's very different actually from the US. In the US, genetically modified foods have been accepted for quite some long time. In this country, it's very different. 43% of people think that GM food is very or fairly harmful. I think they're wrong, but that's their perception. And without proper labelling and giving them choice, we're overriding their autonomy. In the EU, I think it rises to 56% think that it's dangerous. Actually, quite fun, since I have some views on alternative medicines, that 4% of those same people think that herbal products are very unsafe. Now, herbal products are usually not tested. They haven't gone through the rigorous procedures that formal foods and pharmaceuticals have gone through, and yet people think they're safe, and GM crops that have been tested are unsafe. I think there's a mismatch in the logic there somewhere. And you might like to look at this article from a BBC website from a few years ago over GM food in Zambia, where the Zambian government discovered that the maize that was being imported was a genetically modified form of maize, and therefore banned its usage in a country where there was a desperate need for that particular food. And I ask you to think about the ethics of that as to what was the greater good or the greater wrong in that, as the president in those days of Zambia said that GM food is poison and banned its usage. Genetic modification of animals maybe is a more contentious thing yet again. I like that quote from Animal Farm, as George Orwell wrote. The creatures outside look from pig to man, and from man to pig, and from pig to man again, but already it was impossible to say which was which. Now that's in the context of the book Animal Farm, not genetic modification, but some people fear the same sort of thing. I do like Doctor Who, and I don't know if you saw those episodes of Doctor Who a few years ago, where we had the Daleks in Manhattan, followed by the evolution of the Daleks. So we had human-Dalek hybrids, and we had pig-human hybrids, and the ethical questions that those raised. And actually, if you remember that episode, those of you who saw it, is that people were tested, and the more intelligent people were destined to be hybridized with the Daleks, and the less intelligent ones were destined to be hybridized with the pigs as a slave class. And I think that's some of the fears that people have about genetic modification, that it's going to turn us into animals and animals into people, and it's all going to be rather repugnant. Let's look for a moment about how we do genetic modification with animals. This is not to do with stem cells. I'll come back to stem cells in a moment. It's very simple. We simply take the DNA for a particular trait, a particular gene, and transfer that either into a newly fertilized egg or into an oocyte, and then just simply allow that to develop. Some of that DNA is taken up. It's taken up usually randomly into the genome, which means that when it's finally going to be expressed, you can't control the levels of it. It's empirical, deciding which clone to take, which expresses the particular gene you want in the right amounts. You then take this, develop it to an embryo, something like in vitro fertilization. It is applied, it's inserted into the uterus of a potential mother, and the net result is your genetically modified animal. I'd point out that at this stage here, where the embryo develops, has a very low success rate, much lower than the success rate of IVF, and we'll come back to that as we look at some of the risks associated with genetic modification through stem cells as well. There is, of course, the other route I've hinted at through stem cells, where you put the DNA into a stem cell and add it back to an embryo, or allow it to grow into an embryo, and I'll come back to that later. Why would we want to do it? Why want to modify animals in any way? And there are a number of reasons that, again, you can decide which ones of these are worthwhile reasons and which are trivial. Medical reasons, and we'll come to some of those later on. We may want replacement tissues. We may want replacement therapies. Research issues. We begin to understand how genes work by putting them into another animal, into another species, and to see what effect it has on that. We may be able to knock out genes, as well as moving genes around. We can take genes away and say, can that mouse survive without that gene? You begin to learn something about the role of that gene in there. There's curiosity. Now, what if? There are lots of things that I'd be fascinated to know the answer to, which actually the process of finding that answer out would be immoral, or rather unethical. But there's something in all of us that we have that curiosity-driven approach. The child wants to know, well, if I put my finger in there, even if they've already been told that they mustn't do it, we just love to find out, don't we? Egos are tied up with this as well. Scientists are no exception in having overinflated egos, and they want the prestige of being the first to discover. There are commercial reasons for doing it. Drug companies, agricultural companies, all sorts out to make money. Now that's the way the world works. If we're living in a capitalist society, we can't choose to do away with the technology just because money's involved, but nonetheless we bear that in mind as well. And finally we do some things just because they're fun. Again, a little bit like the curiosity, it's nice to know what happens. Let me give you a few examples. Quickly flick through some examples of genetic modification of animals, and you think what you think of this. So here's Alba the bunny. This is a rabbit that expresses a protein called green fluorescent protein. It doesn't do the rabbit any harm at all, but it means that put under a UV lamp and the bunny fluoresces green. And Eduardo Ketch talks about this as being an artistic thing. It has no use at all, but it's the final here is a transgenic animal that challenges the notion of purity. We have the Enviro pig. It's a pig that is genetically modified to contain the enzyme phytase that breaks down phytin and therefore changes the amount of phosphorus that's excreted by the pig. It's environmentally a lot more friendly. The pig utilises the phosphorus in its diet a lot more efficiently. And people ask the question with this, is it safe? Could it jump to another species? If it did, would it matter? Will it affect the pigs? Is it toxic? Is it allergenic? And all these things can be tested. I suppose for those of you who eat bacon, the question at the end of the day is, would you eat the bacon that comes from the pig? What about the next one? Glowfish. These are, again, genetically modified fish that have different colours. They've got different pigments in them. They're originally produced with the idea of using them for testing for water pollution. They don't seem to be any use for that at all, but they're used as an aquarium novelty. The company that make those, Glowfish fluorescent fish, that on their website they do list some of the ethical principles that they apply as they market these things. So they do say, to help ensure that we use this technology appropriately, we dedicate ourselves to the following guiding ethical principles. And risk is the first one, environmental safety, humane treatment of the fish, advancing scientific research, and open and informed discussion. Seems to tie with a lot of those ideas of not inflicting pain, allowing autonomy, being just. Other examples, Cupid and Diana were sheep that express human serum albumin. So it's a human protein expressed in their milk. That means that extracting that, and that's used as a blood product, is therefore very useful. We have spider goat, which is a goat. It's just, physiologically it's a goat, but in its milk it expresses the spider silk protein. So it's a way of making a form of silk that's extremely strong as Kevlar. Getting a little bit more contentious, we have oncomouse. Oncomouse is a genetically modified mouse that will inevitably develop tumours. And it's used in experimental labs for being able to test anti-cancer drugs. That clearly is inflicting some form of a pain on the mouse, But is that justified in terms of the benefit that will come to humans through being able to develop new anti-cancer drugs? What about monkeys that are genetically programmed to suffer from Huntington's disease? A terrible disease for humans. It's a late onset disease, typically affecting people in their mid-40s. It's genetically carried. It's a dominant genetic disease. And these monkeys have been genetically modified so as to be able to mirror that disease and to use for testing for new treatments. What effect is that having on the monkey? Those monkeys there are green in that picture simply because as a marker, green fluorescent protein has been put in with that as well. What about transgenics for what we call xenotransplantation? There's a lack of organs for transplants. There are far more people needing organ transplants than there ever will be the possibilities of organs available. Should we be able to use the animal organs as spare parts for us? And particularly pigs are the one that have been muted on that. Should we be able to use, change the genetic makeup of a pig so that a pig heart is less likely to be rejected when put into a human? And there are examples of these. These pigs here have been knocked out. One particular gene has been knocked out so that they're less likely to be rejected when put into humans. People suggest that while eating pig meat might be natural, using pig organs seems to be meddling with nature. Does it threaten the integrity of the human body? We're built the way that we are, and to tinker with that somehow seems to be messing with evolution and with nature, with unknown consequences. Risks again come into this. We don't know, are there viruses within pigs that are put into humans could be the next HIV, for example. They're not called PERVs. These are porcine, pig-like, endogenous retroviruses. And there are objections because actually pigs are different to us, and that maybe pig organs won't be so useful to us as we might think. In terms of what it does to the pig, I suppose the question is if it looks like a pig and acts like a pig, then I would just simply still call it a pig, and the genetically modified pigs still look and act and behave just like pigs. There doesn't seem to be any damage to the pig. Would there be an ethical problem to growing a human heart in a laboratory if it was technically possible to do so? And I think the answer probably would come back, no, there isn't an ethical problem with that. Is there then an ethical problem with growing a human heart in another animal? And are we then, is there an ethical problem? Or are we suffering from the yuck factor again? But things can get a little bit more complicated. We can make chimeras, we can express cells from humans in other animals by injecting stem cells into them. If you take that at the early embryo stage, you can make a chimeric embryo, which consists of some mouse cells and some human cells. We could do that. It's not allowed. That is not the form of hybrid embryo work that is permitted. What happens is when that embryo develops, it becomes a mosaic. Some of its cells are human and some of its cells are mouse. It will probably still look like a mouse. But these germ cells could be either mouse or human. So in other words, when that mouse, should it mate, it's possible that something that looked like a mouse could have human germ cells. And you could have therefore a human oocyte and a human sperm that therefore had a human offspring, even though the parent was a mouse. Does that sound repugnant? It does to me. What would happen if it's, alright, that's bizarre if you say it's a mouse, but what happened if that was a gorilla? Another primate? What about the human Z? I actually, scientifically, it'd be fascinating to know what happened if you crossed a human with a chimpanzee, because it would tell us lots of things about what consciousness is. It would tell us the differences between our physiology and that of a chimpanzee. But I think you and I would agree that it's wrong, fun though it might be. What percentage of human cells is needed to make something human? 99 or 98.5% of our genome is the same as that of a chimpanzee, but nonetheless That doesn't mean to say that we're 98.5% chimpanzees. We're 100% human, and they are 100% chimpanzees. What percentage of cells would we need to transfer before an animal stopped being one kind and moved to another? Let me change tack just a little bit and talk a little bit about stem cells, since then we hear lots about those in the media. And apologies for those of you who are already totally familiar with what stem cells are. If I go through some simple biology, here we have the fertilized egg, newly fertilized, single cell that begins to divide. So 1 cell to 2 cells to 4 cells to 8 cells. So we get to this 8-cell stage typically after about 3 days. It's a ball of cells. Those cells are all identical. You can remove one of those cells and test it. In fact, that's what's done for lots of genetic screening, to be able to see whether the embryo at that stage before IVF suffers from any particular genetic abnormality. So removing one of those cells does no harm to the embryo at all. It recovers, it continues to grow quite naturally. At this stage, the embryo can split, so it can divide completely into two separate balls of cells, and that's where identical twins come from, or triplets, or however many it might be. So this itself, this ball of cells, although it's destined to become a human, not every cell in that would do, and it's not necessarily destined to become a single human. This then continues to divide, and after 7-ish days we get this form here, where we call the blastocyst. And we have on the outside layer of cells here what will go on to form the placenta, and the inner cell mass in here. And there's that inner cell mass that becomes the stem cell that become part of so much debate. If we think about this original single cell is what we call totipotent. That can make every cell in your body. I still find it fascinating, even though I know a little bit about developmental biology, how something as complicated as you and I can arise from just one original cell. Now the science is there, but it's still fascinating and almost awe-inspiring. Once we get to this stage here, these cells on the outside are already what we call differentiated. They're destined to become placental cells. And the ones in the inner cell mass can become anything except placental cells. So they're what we call pluripotent cells. They can't do everything, they can do most things. So it's possible to take those cells out from the blastocyst, to separate these and culture them. So you can get from those few cells, by preventing them from differentiating. Normally they'll differentiate into all sorts of different cell types, and in the human body it's reckoned there are about 200 different types of cells, fulfilling different functions, expressing different gene products. And it's possible, by controlling the media in which these stem cells are cultured in, to stop them from doing that. They simply remain as an undifferentiated cell that we can then take later and cause to differentiate into a heart cell, a muscle cell, a bladder cell, or all those sorts of different possibilities. So this sounds quite nice in terms of therapy. We can take these cell types and use them for regenerative medicine. Of course, the ethical question comes as to the source of these embryonic stem cells. Their source has to be from embryos themselves, hence embryonic stem cell. And in order to do that, you therefore destroy the embryo. That's the big ethical question, is are those embryos human or are they not? Most of them come from embryos that are surplus to in vitro fertilization, which generates lots and lots of embryos that are stored for a number of years and then discarded with parental consent. And they could only be used in science with parental consent. The question is this stage here, is it human or is it not? And that's the ethical question. And there are a variety of different views on that. From the view that says that the early embryo is a human person, and that destroying that embryo is destroying a human. And so people are as opposed to embryo research at that early stage, just a ball of undifferentiated 8 cells, as they are opposed to, say, abortion. And that would be the thoroughly pro-life opinion. There are those who take the opinion that the early embryo is not a human person, but it has the potential to become one. And that until the embryo is implanted within the uterus, then up until that stage it is not human. It has the potential, but it's not human itself. And of course there are those who say the early embryo is just a clump of cells. And actually there's nothing special at any of those cells. I've already said they all look the same. Now you've got to make your own mind up on that. Different religions take different views on that. The Catholics, of course, take a pro-life approach. That to tinker with an embryo is to tinker with a human being. A Jewish religion comes much further down this list here. That it doesn't become a human being until later on. I forget the number of days. Actually, I think in Jewish theology it's different from a male to a female as when it becomes a fully human being. So within different religions there is a spectrum of views as to when you can and when you cannot do things with the embryo. Now, assuming we can, we have the possibility of therapeutic cloning through these embryos. This is not generating new people, this is generating new tissue. So we go through the procedure of getting these embryonic stem cells. We take out the nucleus from an ordinary cell, inject into that the nucleus from another cell, and grow up these embryonic stem cells. They can come through them, we harvest them down here, and then at the various harvestings, then you can make them go into all sorts of different cell types for use in regenerative medicine. And that might be of great benefit. For those who suffer from Parkinson's disease, where dopaminergic neurons in the substantia nigra are destroyed. For those who suffer from spinal injury, and you've seen the cases of that, could we cause neurons to regrow? There's a thing in the media on this very week of those who were able to generate cells in the ear, the fine hair cells from stem cells. For treating heart disease, getting the heart muscle to repair itself. For generating all sorts of new organs. They could be used in basic science for understanding how cells differentiate and understand genetic abnormalities. And they could be used for all sorts of pharmaceutical testing, so you test a drug on a cell rather than on a human being. Now you have to weigh up the ethics of that as to, are these things human or are they not? And what's the greater good in all this? We have to destroy something in order to create something. I can't make that decision for you. Of course, the issue becomes a little bit easier if we talk about adult stem cells. It's possible, although harder, to extract some stem cells from, the word adult is actually a misnomer, but it's that from a fully formed human being, whether that's a child or an adult, and use those. There are all sorts of problems with that because, of course, they're already partially differentiated. Compare that, though, with the reproductive cloning that people talk about. That's generating new human beings. So this time we'll get through a clonal embryo and now insert it into the uterus so as to be able to get a whole line of clones. And that essentially is what was done with Dolly the sheep, which was really the first cloned animal. So we pointed out, I've already said before, that this sort of technique is very, very inefficient. Dolly was one of 277 experiments, and that was the only one that survived. Many of them died very early on after implantation, and the vast majority didn't even implant. They were not viable. So as we talk about any form of cloning, we have to emphasize, at the moment at least, risk that you'll be throwing away 277, and of those 277, you might find some of the successful ones will be highly deformed. So what about the health of the individual that results from this? Dolly the sheep died after only 4 years. Typical lifespan for sheep, I'm told, is between 7 and 8 years. Some of you may know more about that than I do if you come from a farming community. The cells that grew in Dolly were already old. And there's some arguments as to whether it was bad for health. But of course it raises the questions of designer babies, and if we cloned babies, what would be their worth? Would they be valued as a commodity? Would they be loved as children, or would they just be rather like machines and toys. The arguments for that, I think, are quite strong, but they're no more so actually than why a lot of parents already have children anyway. It is an extension of their own egos, and they have real expectations of what those children would be. Most parents would hope for a highly intelligent, very sportive, blue-eyed, black-haired child, grey-haired child. So they hope for the best, and is cloning a means of achieving what they only hope for, rather artificially? I put that out rather provocatively, because actually I don't think that's a good excuse. For a moment about the use of animals in of human cloning again. Those arguments for and against. Against, if we could clone humans, would ask questions about our uniqueness and identity. But who are we? If you're simply a clone of somebody else, it does ask a very deep question about who we are. The but side of it says, but actually identical twins are genetic clones, but they're not identical. Beware of deterministic assumptions that we are nothing more than our DNA. A clone will have identical DNA, but we know that identical twins are different, they're different personalities, they're different people, so beware of that argument. We are more than just the sum of our genes. People talk about the effects it has on freedom, so that would clones be simply puppets with inevitable traits? Comes back to this idea of parental expectation. But of course our development depends on all sorts of other factors, and we've already said that parents already have expectations of their children. What about the talents of an embryo? If we could have a child that was specifically very athletic, going to be a good muscle tissue, would that be an authentic human being? The other side of that is it will still need an active role. You could have all the genes necessary for giving you good muscular activity, and you could be a couch potato and sit in front of the television all day, and you wouldn't be an athlete. So there is still personal autonomy in this. And then, are we looking as humans as simply commodities? And again, I think some of the arguments against that come back to rather deterministic ones. Let me skip ahead several slides. Right, there's lots I could say. Sorry, that's the slide I wanted. Is it right or is it wrong to do it? The danger is that we see some of the risks, we see some of the ethical problems, and we freeze because of that. And we say we're therefore not going to do anything. And I rather like this for a quote from Francis Collins, who says that to say that genetic engineering is unacceptable across the board because of its potential for creating some ethical dilemmas is the most unethical stance of all. It's to say basically, here is a powerful approach which could alleviate human suffering, but we're not going to do it because we're worried about the misuses that might occur. I find that completely unacceptable from every point of view. And Francis Collins, former director of the Human Genome Project, and a very outspoken Christian in the US. What about another ethical dilemma? Human screening, DNA screening. It's possible to get lots of information about your genes for, I think, a matter of only a few £100. You can get a DNA profile, which is essentially what you contain in terms of, I think it's about 500 or 600 different genes as to what your susceptibility is to that. If you do that, and you seem to be susceptible to a particular disease, is there a genetic counselling then to tell you what to do? I'm told that in various parts in the US where this genetic screening is quite common, there are more companies or more genes that can be screened for than there are genetic counsellors to be able to tell you what to do with that information. Information by itself might be fun, but it can be absolutely useless. Who then has access to that data? Who owns the data? Not just allowed who has access to it. And if we use this prenatally, what again are we going to do with that data? Come back to some of those questions. If we do genetic diagnosis prenatally, so before implantation we could do that, you could discover that the early embryo had a genetic disease, would you discard that embryo? Would it be ethical then to go ahead and implant that? If you discover it post-implantation, when the fetus is developing, again you can do the genetic tests. If you discover a serious genetic disease, and then we might argue what is a serious genetic disease, would that be sufficient for asking for an abortion? The genetic screening that is done in advising couples as to whether they should marry and have children or not. Amongst the Ashkenazi Jews, who suffer a lot from a genetic disease called Tay-Sachs syndrome, which is a recessive disease, which if both parents are carriers, there's a 1 in 4 chance that a child could have that. There is a means of screening amongst the Tay-Sachs, amongst the Ashkenazi Jews, for seeing whether a couple about to get married actually are both carriers of that, and if they both are, it's suggested that maybe they ought not to marry, and if they did, they ought not to have children. A similar thing with the blood disease thalassemia amongst particularly the Greek Orthodox community in Cyprus, again warning the possibilities of what could go wrong. But does that give rise to some form of genetic discrimination? If you know that you have a particular disease or susceptibility to a disease, are you going to be discriminated against? Comes back again as to who's going to know this information. Should you know it? Should your parents know it, or your children know it? Because that says that if you've got the gene, they may have it as well. Should your GP know it? Are they going to look at you differently when they treat you if they know that, they're going to get heart disease? Should your insurance company know you've got that disease? Your insurance premium, if you've got a predisposition to heart disease, is going to be very different to if you're not going to have heart disease. Should your employer know about it? Because if your employer is only obviously going to take people on and train them up, if they're going to have you for a good number of years, as a possibility. It is possible now even to go personalised genomes, not just to have those single genes susceptibilities, but actually to know the sequence of the 6 billion bases that make up your DNA. And the first of these done for fun really was James Watson, done a couple of years ago. And even within his genome, there were 20 serious genetic abnormalities. That's it. So that if he had a child with somebody else who had the same abnormality, there was a serious chance that the child could be seriously deformed. Even within his genome, he has chosen not to know which form of the gene ApoE he has. Now, ApoE is a gene that gives us increased susceptibility to Alzheimer's disease. I was at a scientific meeting where a test for that was announced. Somebody had developed a genetic test for whether you were a carrier of the ApoE gene or not, and there were 300 scientific delegates there, and we said that if any one of us wanted that test, they would do it to us for free, and nobody wanted the test. Quite simply because what would you do with the information? There is no treatment for Alzheimer's disease. Knowing you're going to suffer from it actually increases your worry rather than decreases it. There's the whole area of pharmacogenomics is related to this. The ways in which we respond to drugs are different. Some of us are allergic to one particular drug. Some of us have one side effect. Others have another side effect. Knowing your genome will be extremely useful in developing personalised medicines. So there's this enzyme in the liver responsible for inactivating a lot of drugs, but about 10% of Europeans don't have it. It actually means that about 10% of Europeans are non-responsive to codeine because it's metabolized into an active form. Herceptin, we've already talked about, used for treating breast cancer, only works for those who are carriers of the HER2 gene. Genetic testing will say who would benefit from that drug and who wouldn't. So we get fantastic benefits from personalised medicine of appropriate use of drugs, reduced cost, improved safety. But the downside of this is it's going to lead probably to some orphan diseases. If you happen to be the person who suffers from a disease that you can't take a particular drug, is the other drug company still going to invest in finding treatments for that small number of people? What if there are no drugs left for your particular disease? And it does actually have the other side of it, of what lifestyle choice you're going to have. If you know that you're never likely to suffer from heart disease, then you might as well pig yourself at dinner this evening. Whereas if you know that you are going to suffer from it, you probably ought to take it easy. Similarly, if you know that you're going to be especially susceptible to lung cancer, then you will stop smoking. Whereas if you know that your genetic profile is such where it doesn't really matter, you'll carry on. And there's all sorts of ethical questions within that. I want to finish just with one other ethical issue that's rarely talked about, and that is the ethics of scientists. That we are humans too, and we suffer from all the same human traits that anybody else suffers from. We have large egos. We spin things. When you hear a report in the media about a new scientific development, ask, how did the media get to know about that? Sometimes it's because the scientific journals tell the media, sometimes it's because the university press office tell the media, and sometimes it's because the scientists themselves tells the media, because their grant might be coming up for renewal and they want a little bit of publicity. New cure for cancer, you see in the press. And it rarely is a cure for cancer. It's the discovery of a new gene that might lead on to a new therapy for cancer 20 years down the line. Funding is tied up with all with scientific endeavour. How do we get funding to be able to do the science, whether it's from research councils or whether it's from industry? If it's from industry, is it dirty money or is it not? I don't think it is necessarily, but are there strings attached to it? People's careers, scientific careers, now depend entirely on being able to raise money. In some universities, my own included, there have been redundancies because people haven't had research money. So the drive to be able to get that research money, and sometimes to be able to tell lies, so to be able to get that research money, because your career might be on the line, can be a temptation. Here's a quote from an Australian Christian journal that talks about the ethics of being a scientist. I'm getting close to the end now. So the scientific enterprise depends critically on the honesty of the researchers. I think we would dispute that, would we? The ethical requirement is not deducible from science itself. Historically, it's derived from Christian ethics. Now, he broadens that to say there is no reason to quarrel with those many scientists who derive similar ethical standards from other religions or philosophical traditions, of which several could be cited. But what could be expected from those who follow recent philosophies which assert that ethical standards are entirely relative to social structure or entirely personal or subjective, or that there is no truth out there to be found? One story is as good as another, and he adds one cannot be very hopeful. Or Max Perutz, and I have no idea what Max Perutz's faith stance was. He was responsible for discovering the structure and atomic detail of hemoglobin, a fantastic scientist. It said scientists may not believe in God, but they should be taught they ought to behave as if they did. And somebody's watching over you to make sure you tell the truth. For most part, research will be ethical if researchers believe the truth must be told no matter what. Follow the truth no matter where it leads you. But what if scientists stop believing that and stop behaving that way? Science is only reliable to the degree that scientists are honest and truthful. and this came from the British Medical Journal. Many researchers think that a high IQ, and arrogant enough to think that scientists have a higher IQ, goes hand in hand with high moral values. This correlation is, of course, absolute nonsense. Scientists are human too, be very intelligent and also very devious. And we've known of some cases over the years of scientific fraud. I'm an editor of a scientific journal, and I would say once or twice a year I see evidence of scientific fraud, people manipulating data for one reason or another. The most recent case that made the big news is the Korean, Hwang, who fabricated his results into stem cells, maintained that he had generated, I think it was 11 different lines of stem cells, and it was all a fabrication and all had to be withdrawn. Lysenko in the Soviet Union, back several decades ago, fabricated results that had profound implications on Soviet agriculture, making his results up for political rather than scientific reasons. And of course there's sometimes the joke scientific fraud, rather like Piltdown Man. So scientists too are open to abusing the system. And this is, I think, my last slide. One thing that as we apply for grants now, we have to, in order to show that your work is worth funding and it's not completely off the wall, you have to give preliminary data. You have to show that it's going to work. And I wonder how many scientists, in fact I don't wonder, I know, how many scientists put in their proposal things that they will do, where actually they're lying, they've already done them. And they're asking the Research Council to fund what's already been done. It's on the edge, isn't it? You can see why they're doing it. The scientific result is not going to be in question, but the process by which scientists sometimes are required to tell half-truths in order to be able to get funding, I think is highly questionable. And so this one talks about anecdotal evidence for this. Unless researchers follow this less than honest procedure, they will not get a grant. And when the CES system compels such dodgy procedure, one should not be surprised if some corners are cut elsewhere as well. Are we breeding scientists whose egos are larger than their integrity? So it's just the same about let me close by just giving you some, and they're on the sheets, some other resources. I meant to suggest this one was on the bookstore. This is John Bryant's book with a couple of other people, John Searle, and Linda Baggett-Lavelle. On Introduction to Bioethics. There's another book by John Bryant on the bookstore, which I highly recommend as well. And this subject moves very rapidly, and therefore actually books are often not the best source of material. And those are 4 very useful websites that dip into, some particularly, the BEAP one is particularly designed to bring ethical issues in education to the fore, as to in terms of higher education, particularly the Bioscience Higher Education Academy, but there's lots of teaching resources within that as well, and those websites should be on the handout that I gave. Sorry, I think I've gone over time. Thank you for your patience, and it's nearly dinner time.
Thank you very much, Keith, for that fantastic overview. We are going to take a few questions now. We're going to have a break before dinner, and because we do have the panel and there's a chance to ask more questions for Keith through the question box. But let's just have a few questions now. Yes, please.
Yes, I was wondering what your opinion was on using the implantation genetic diagnosis to select a sibling in order to provide a donor for an already existing sibling.
That's a really thorny issue, because it's generating a life to save a life. There would still be the selection of the child would grow. It would still often be another perfectly valid, loved human being. So in that sense, I don't see any problem. But if the child is there simply as a commodity, as a means of a spare part, as a means of extra tissue, of spare cells to save another child, then you have to question that. This is where it becomes very personal, because I think then it depends on what's the environment in which that saviour sibling, if you like, is going to be reared. And you can't legislate for that sort of thing. So I would almost say that has to be done on a case-by-case basis. That's my own view. I'd say I'm raising more questions than giving answers.
Yes, we go there.
Are the ethics associated with biological research any different from any other field of scientific research? I think when they're investigating quantum mechanics at the start of the 20th century, that leads ultimately to a bomb being dropped on Hiroshima. Is any of this any different to good science being reflected?
Some of it is slightly different. I mean, there is, and I think governments consistently get this wrong, is the distinction between science and technology. And governments want to fund technology, they don't want to fund science. Technology is the appliance of science. Science is simply discovering how things work, how the world behaves. And in terms of the physics, say the atomic bomb, we're talking about the technology being used. I think there are aspects of biology, though, where the very process of being able to find the scientific information out is itself has ethical questions. So my question, a hypothetical one, of what would a cross between a human and a chimpanzee look like, would give a scientific answer, but there's actually the process of doing it would be unethical. So I think it's not just quite technological within the biological sciences, where some of the examples within the physical sciences are simply the use of a technology, an appliance of a science, rather than the science itself.
Yes, question.
Do you agree with Steve Jones that ethicists have no business telling geneticists what to do?
No, I don't. I think quite the opposite. And frequently, actually, a lot of biological experiments now have to be submitted to ethical councils, ethical committees before they can be done.
You think it's the right way around?
I entirely agree it's that way around. It may be sometimes over the top and over cautious, but I think society is only going to accept the scientist as somebody of benefit to society and not as an ogre if they're subject to public scrutiny.
Unaccountable.
And totally accountable. And the process of autonomy comes into that. If society says, no, we shouldn't go down that way, I think there comes a point where, rightly or wrongly, the scientist has to stop.
Thank you. Yes, please.
Yes, it must be about 10 years now that human insulin has been got from bacteria. And in that time,
Longer than that, actually.
sort of longer than that, right. In that time, I've never heard of any new patients having huge adverse reactions to it. One could perhaps draw a conclusion that it's safe. Why have we not used this to go on the offensive with the GM food issue in terms of the risk to society, when it seems there is this marvellous resource out there and we're afraid of perhaps the anti-GM campaign?
Yes, I think, a bit provocative, I think a lot of the anti-GM campaign in this country and in Europe has been political rather than scientific.
But why haven't we gone on the offensive to say, actually, we've got 10, 15, 20 years worth of data out there, it is safe,
Yeah.
and to presume that it's not safe?
I mean, even more so than the insulin, which is, of course, a bacterial product now. The GM foods, genetically modified maize has been on the market in the US for 20 years. 20 years, do I mean 20 years? I think so. But certainly there's a lot of data, and I'm not aware of any toxicity data with that. And so you're right.
Yet that is still an argument that's used in Europe, is, well, we don't know what the long-term effects are. And that's patently false. We do know what the long-term effects are.
We know the long-term effects of some of these things quite clearly. Yes. So, again, it's what the media like to pick up, it's what people hear, it's people's misunderstandings sometimes, and their lack of knowledge of the science. It is, and I forget where I read it, and it was from a poll, must have been taken 10 years ago, to show people's ignorance of science when asked about DNA, thought it was only genetically modified organisms that contained DNA, whereas ordinary organisms didn't. So there's that basic mismatch in the general public's understanding, and it's the responsibility of you as teachers to be able to make sure that the coming generations have that basic scientific understanding to be able to cope with some of these issues and not rely on the sort of inflammatory statements that are put in some of the gutted press, which aren't scientifically based, and to use data just exactly as you say.